Okay, this problem can be a little tricky if you're not used to figuring questions like this out backwards. But this is the best way to learn. Instead of giving you all the numbers and having you plug them into equations, it's better to have you work backwards like this because you really get to see the patterns that emerge. So let's take the easy part first. We know that if we have no lactase non-persistent, no lactose intolerant people, 0%, that represents our homozygous recessive condition. So we gotta have 0 in that group. There are no people in our population that have that genotype. Okay, so now that means we gotta split 1,000 people between these two genotypes, homozygous dominant and heterozygous. The only scenario I can calculate that gives me this particular allelic frequency is 500 in each group. So remember, both of these are lactase persistent. So together, that's everybody, 100% are lactase persistent. But we need a number that gets our allelic frequencies to 0.75 for the dominant allele and 0.25 for the recessive allele. So if we go to our equation here and set it up to work backwards, 0.75 equals, we have the total number of alleles. So how many total dominant alleles are we dealing with here? This would be X equals 1,500. We've got to have 1,500 dominant alleles in here. We do the same thing with the recessive allele, and we got to have 500. Well, if we have to have 500 recessive alleles, the only way I'm going to get 500 is in this heterozygous condition because I don't have any homozygous recessive. So that means to get 500 recessive alleles, I've got to have 500 heterozygous individuals so that I get 500 of these. 500 heterozygous individuals then makes up 500 of my dominants as well. I need 1,500 total, so subtract 500 from 1,500. I need 1,000 more alleles. 500 in this box takes care of 1,000, right? Because I get 500 of both, 500 of the first dominant and 500 of the second dominant. Easy, huh? Or hard, depending on your perspective. I really hope you understand this. If you don't, if you have trouble, it's okay. There's not a lot of this course that's going to deal with too much allelic frequencies, but I do want to focus on it a bit more for the rest of the lesson. If you can really get a grasp of these allelic frequencies, you're going to have a whole new appreciation for understanding gene flow, genetic drift, and looking at huge populations of individuals and seeing how alleles travel through and can be shaped by evolution.